Well, thanks everyone for being here virtually today. So today I'm going to be talking not about a new bioconductor package, but about my work to contribute to Splatter, an existing bioconductor package for simulating single cell data. So single cell data, or the amount of single cell data has been dramatically increasing the past few years, especially starting around 2015. And unsurprisingly, the number of bioinformatics tools available has been increasing also, with over 700 tools available today. Now, when you're trying to decide what type of software or statistical method to use for an analysis, you want to know how the tool's performance compares to others. But performance is really hard to measure using real single cell data because there's still a lot of unknowns about single cell genetics. So one solution to this is to try to simulate data where the ground truth is known. So there have been quite a few tools and methods proposed for simulating single cell expression data. And in 2017, Luke Zappia and Alicia Oshlak published Splatter, which implements a number of those models, including their own model called SPLAT. So SPLAT samples gene means from a gamma distribution and then samples the counts for each individual cell from a Poisson distribution. And those distributions are parameterized from real single cell data. So with SPLAT, you can simulate really cool complex data sets like these, where group effects might indicate different cell types, or path effects might be different steps along some kind of differentiation trajectory. It also allows you to simulate batch effects, so you can have more complex experimental designs. So Splatter is a really fantastic tool. It's been cited over 200 times in just a few short years. But one of the issues is that as single cell sequencing becomes cheaper and higher throughput, more groups are generating single cell sequencing data for whole populations because they want to be able to perform population genomic studies like eQTL mapping at the single cell level. So just really briefly, a quick recap of Qtl mappings. So quantitative trait loci are genetic loci like this one that are associated with some kind of trait, a quantitative trait. In this case, when we're talking about eQTL, we're talking about the expression as the trait. And there are a lot of different reasons why a genetic variant might be associated with change in expression. And one of the simplest mechanisms is that this genetic variant might be in a region that affects the binding of a transcription factor that regulates a particular gene. So for example, here, having a G rather than an A at this site improves binding of this transcriptional activator, and so it's associated with an increase in gene expression. So that seems simple enough, but these type of regulatory mechanisms are really hard to find in bulk sequencing data. And one of the reasons for this is that the cellular context is really important. So for example, perhaps this regulatory mechanism exists in intestinal cells, but in liver cells, this region might be really heavily methylated. So the genetic variant at this site will have no impact on expression levels. So the promise of single cell data and single cell eQTL mapping is that we'll be able to find these cell type specific or even dynamic eQTL, which are only present at certain developmental stages. So this is what motivated us to develop SplatPop, which is an extension of the SPLAT model that allows you to simulate single cell eQTL data, or sorry, single cell expression data for an entire population. So SplatPop has two separate phases. The first phase is the simulation of gene means for all individuals in a population. And then the second phase is the simulation of the single cell count data. Now, the second phase is essentially just a wrapper around Luke's SPLAT model. So the major contribution of SplatPop comes in this first part, which can be broken down into these 4 steps, which I'm going to go through in more detail now. So the first step is simulating gene means. So the gene mean for each gene is sampled from a gamma distribution that's parameterized from real population scale RNA-seq data. And when we look at real expression data, we can see that the variance in gene expression across individuals is related to the gene mean, where lowly expressed genes like these ones down here have greater variance. So the next step is that the coefficient of variation is sampled using parameters that are estimated from genes in the same expression bin. And then finally, to allow users the ability to manually control how similar different individuals in their population will be, we allow them to add this parameter which will scale the variance distribution's rate. And closer to the end of my talk, I'll show you an example of what that looks like. So the second step is that after we have these means sampled, what we want to make sure is that they look like what we would expect from single cell data. And we do that with quantile normalization, where the sampled means from each individual are quantile normalized to fit the distribution of real single cell data. So for example, here's the distribution of sampled gene means for all genes from one individual before quantile normalization. And these are sampled using parameters estimated from bulk RNA sequencing data. But then after quantile normalization, using parameters estimated from two different single cell technologies, we can see that we get a much tighter range of expression, which is exactly what we would expect to see for single cell data. So now that we have our data looking like single cell data, the third step is to apply eQTL effects. So first, the user can decide what proportion of genes they want to have eQTL effects, and they can also decide or put some parameters on the genetic variants that are assigned to each of these genes, like if they have to have a certain minor allele frequency or be a certain maximum distance away from the gene. And then finally, the effect size of the eQTL is sampled from a distribution parameterized from real eQTL mapping data from GTEx. So this is just a really quick schematic of how those eQTL effects are actually incorporated into the gene means, where the number of minor alleles that an individual has and the effect size that's been sampled kind of influences how big the eQTL's effect is on that gene's mean. So this approach for incorporating eQTL effects into data has already been used to simulate bulk eQTL data, but it wasn't implemented in reproducible software. So we've made that available here in SplatPop. All right, so now at this point, we can actually look at some simulated genes. So here I'm showing the distribution of gene means across the population for two different genes with different means and variances. You can see that in general, these are normally distributed, with this peak here around 0 being a result of quantile normalization. If we look at two other genes that have similar means and variances, but that have eQTL effects assigned to them, we can see this more bimodal distribution where some of the samples have higher levels of gene expression because they have one or more minor alleles. Okay, the last step allows you to simulate multiple cell types for each individual in the population. So these are group effects, and they're generated in two different ways. The first way is by assigning a certain subset of the eQTL as group specific. And that's basically just done randomly. And then the second step is to apply eQTL effects, where genes are differentially expressed in one cell type for all samples independent of the genotype. And the parameters to do that are estimated from real single cell data using base splat functions. So let's look at some examples. So on top here, we have a gene that has a group-specific eQTL effect where you can only see the effect in cell type 2. And then below, we see a gene that has a differential expression effect, again, only in cell type 2. So the output from all of this is a matrix of gene means for each gene, for each individual. And this is the input to basically the wrapper around SPLAT. All right, so these are some final simulations. So these simulations were generated using parameters that were all estimated from real data. The single cell parameters were estimated from iPSC cells from the HipSci project, and the population parameters are from GTEx. The genotypes are from the 1000 Genomes projects, and then I simulated all of the genes on chromosome 2. So here, each color represents a different sample, and then each dot is a single cell for that sample. And by tuning the similarity scale parameter, I can either generate a population where the cells are more similar to the other cells from the same individual, or where we get a better mixture, where this population or the sample effect is much less dominant. There's a lot to see in this figure, but the main point here is that with SplatPop, we can also simulate whole populations with group, batch, and path effects. And importantly, all of these effects are really tunable. So you can make them as subtle or as obvious as you need for your particular case. And then finally, I'm going to end with just showing you a little bit of a teaser about how we can use SplatPop simulations for benchmarking eQTL mapping methods. So what I'm showing here is the performance of eQTL mapping using the simulated gene means, which is essentially just bulk simulated bulk data. So here you can see that we were able to identify over half of the eQTL with a pretty low false discovery rate. And then we can compare that to doing eQTL mapping with aggregated single cell data. And we can compare different aggregation methods. Like here we have using the sum versus using the mean. And you can see that there is a little bit of a decrease in performance of eQTL mapping with single cell data, but that decrease is much less using the mean. And we keep a very similar FDR. And really interesting, we actually see that we're able to increase the correlation between the estimated and the actual effect size of those eQTL that we are able to map. So with that, I'd like to thank my postdoc advisor, Davis McCarthy, and also Luke and Alicia, who've been really helpful and supportive in getting SplatPop incorporated into Splat, and then our collaborators on the benchmarking analysis. And if you are excited to try to use SplatPop yourself, hopefully you'll only have to wait a few weeks as we're trying to get it out in this next Bioconnected release.
Thank you so much for a wonderful talk on Azodi. So any questions? Let's see if there are questions. A lot of single data about blood. So the generate to solid. Let me see. Yeah, I think that there's one for you.
Oh, I see.
Oh.
Yeah, so the SplatPop approach is really generalizable. So all of the parameters in it, there are a default set that are kind of just sensible defaults to start with. But if you have a specific type of data or a specific tissue type that you want to work with, and you have a little bit of data from that particular tissue, then you can use that to estimate the parameters to make the simulations look more like what you need. You consider.
I have one question.
Yes.
Do you think a median, you use a median will help increase the sensitivity, or you don't think that?
So that's actually, we're trying that right now. This is kind of just the very beginning of that, that analysis. So far, the mean has worked a little bit better than the median, but we're still working on other approaches.
Thank you.